2020 has been a momentous year for science and global health.
It sounds like a mind-bending problem.
Increased understanding and awareness of the interconnections between humans and planet Earth has never been more pertinent.
It feels magical here.
And Irish research is helping drive us towards a brighter, better world.
It's ingenious technology.
This week on 10 Things to Know About, we're looking at viruses. Kathriona and Jonathan meet Irish scientists seeking to help us understand the deadly coronavirus, and I find out about the most abundant and deadly virus on the planet that can actually be used for good.
"2020 will be remembered for one single unprecedented global event, the coronavirus pandemic. Hundreds of thousands have died from COVID-19, and despite recent reports of promising vaccine candidates, the virus will continue to challenge our communities and healthcare systems into 2021.
A successful vaccine will be key to getting our lives back to some kind of normality, and thousands of scientists across the world are contributing to that effort. I'm in Cork to meet Irish researchers who are playing a vital role in helping us to understand the COVID-19 coronavirus.
Paul Cotter is lead researcher on the Irish Coronavirus Sequencing Consortium.
The coronavirus is a type of virus. It's an RNA virus more specifically. And these are respiratory viruses that cause infections in animals and birds. And they include a common cold, but also more serious forms of viruses that can cause things like SARS, MERS, and obviously most recently COVID-19. And they have their name by virtue of the fact of their appearance. So they have a spherical appearance with these proteins that protrude out, and it was suggested that they resemble the sun and the corona that surrounds the sun.
There are currently more than 150 coronavirus vaccines in development around the world, and key to their success is a deeper understanding of the spread and potential change in the genetics of the virus.
What we're doing is to look closer at the types and strains of the COVID-19 causing virus that are in circulation, and there's a number of different reasons why you might do so. So one is to determine, do vaccines work just as well against the different strains? And if they don't, then what sort of strains do we have in Ireland to make sure we use the right vaccine? But in addition, we can use DNA sequencing as an almost barcoding system for those strains to see where they've come from, whether the virus that's predominant in Ireland might have originally come from Italy or directly from China. So we're leading a national consortium known as the Irish National Coronavirus Sequencing Consortium, and that involves lots of different hospitals and universities right throughout the country.
Fiona Crispy is head of the DNA sequencing lab in Moorepark.
DNA is made up of 4 building blocks or bases, which we call A, T, G and C. And basically those bases can be in any order, which makes what you call your genetic codes.
The order of these genetic building blocks determines the information available for building and maintaining a biological structure, which could be anything from a human being right down to a deadly virus.
How different is a coronavirus sequence to a human genome sequence?
Well, it's much more simple for a start. It's a much smaller genome, much smaller genome, so it's much easier to sequence. But also, viruses tend to encode very few proteins. They rely on the host or the host machinery to replicate and to make most of their proteins, to help them make their proteins. And that's why they need a living host in order to replicate.
Yeah, they kind of walk this life between being dead or alive in a way, don't they?
They're kind of like a parasite. They have to have a living cell in order to replicate and reproduce.
Tell me how you actually go about analysing the DNA sequence of the coronavirus that's circulating in Ireland.
We're using a technology called Oxford nanopore technology. And what's happening there is that your DNA molecule is passed through a little hole in a membrane. And as they pass through the membrane, it causes a fluctuation in a current that's applied across the membrane. And the size of the fluctuation will tell you the sequence of your DNA molecule.
The consortium is working with coronavirus patient samples from hospitals across the country. In each case, when the fragments are assembled, they generate the full picture of the sequence of the virus.
So this is our MinION device, and this is our MinION flow cell. So this little flow cell slots in here into your MinION device, and basically this is your handheld DNA sequencer. So your sample's just loaded in here. You close back up your ports, close your MinION.
That's it?
Attached to your computer, onto which you have loaded your software.
Wow. And off it goes.
Press run.
That's amazing. When you open it up, it looks like the inside of a mobile phone, but it's actually a lab in a chip.
Yeah, exactly. We have a larger version as well called a GridION, and basically the GridION will take 5 flow cells at a time, so we can run far more samples. It's still early days, but what we have seen is that there are kind of 4 lineages, is what we would call them, of the virus circulating in Ireland at the moment. What's interesting is that Cork and Limerick have a lineage that's very similar to the original Chinese virus, whereas the rest of the country has a great-grandchild of the original more than one that's more closely resembling the original.
That's interesting.
Yeah, it's difficult to tell at the moment, but it is fascinating that you can see that, like where the actual strains seem to be coming from.
Scientists around the world have been tracking the spread and evolution of COVID-19 from the very beginning of the pandemic. And most recently, the Irish team have identified a new lineage which seems to have originated in Spain. It was introduced to Ireland around the end of July and now accounts for a high percentage of positive cases.
So all this scientific effort that's been put into this type of research, how will it benefit us in the future?
In multiple different ways. So we're learning lots about vaccine development. All of our different bioinformaticians are talking to one another. And so if there were to be further rounds or outbreaks of COVID-19 or, God forbid, a pandemic of a different nature, we're much better set up now to have a rapid response to those.
One of the most vulnerable groups to be affected by COVID-19 has been older people, and the Irish Longitudinal Study on Ageing, or TILDA, which began in 2008, has the potential to help us understand why.
I've come to the Mercer Institute for Successful Ageing to meet Professor Roseanne Kenny and find out why the TILDA study is so well set up to measure the impact of the coronavirus pandemic and lockdown measures on older people.
Older people are probably one of the most susceptible groups to COVID because our immune systems change so much with age. Even after the age of 40, they start to alter. So that all of the cells, which normally become very, very active when we're exposed to something that's foreign to the body, they're all slightly muted with advancing age.
Tell us about the TILDA study. Why was it set up in the first place?
TILDA is a longitudinal study on ageing. We've been following the same cohort of people now for 12 years. And the value of that is in the 9,000 odd people in the sample, we can actually make generalizations about the population at large. We can then identify early risk factors, early markers, early indicators, and hopefully that will help us to discover new treatments, but also new markers that will modify the aging process, decelerate the aging process.
As part of the rapid research response to COVID-19, TILDA were awarded extra funding to examine the effect of the pandemic on older people.
Dr. Mark Ward is a TILDA Research Fellow at Trinity College Dublin.
The really unique feature of the COVID study is that we have 10 years worth of information that our participants have provided us with. We can track how people have changed over time. So there's a lot of surveys at the moment that are taking a snapshot and they can say, you know, there's a high anxiety levels, depression and so on, but what they can't show is how people's anxiety or depression have changed due to the pandemic.
Participants on the TILDA study are highly engaged. Imelda Fitzgerald is from County Sligo.
Imelda, you're one of the youngest participants in this study. Why did you want to take part?
I suppose my husband and I, we were randomly selected. We thought it was a good opportunity to get involved in it and that there would be encouraging benefits from it. It is good to have tests done and keep checks and make us more aware of the the ageing process and how important it is to keep active and healthy.
So what did they ask of you? What questions did you have to answer and what tests did you have to do?
They covered all areas like from your income, your health, your lifestyle, your mental health. We had blood tests done, eyesight tests, hearing, balance, and then memory, yes, that's always on the questionnaires, the memory tests. And they were good fun as well. So we'd be competing, myself and my husband, to see who did the best on the memory tests.
The study's looking at the effect of the COVID-19 pandemic on an older population. How did it affect you?
My daughter's in Australia, and she was due to come home for a visit in the summer, and she won't be coming for Christmas either, so it is still affecting us. I have grandchildren who live in Sligo as well, and we'd have seen them regularly, but we had to put our distance there. I suppose the restrictions of the 2K and the 5K were fairly tough in the beginning, but we got on our bikes, met our neighbours. I even got on Zoom.
Findings from the study suggest there are a number of actions that older people can take to reduce their chances of getting viruses like COVID or other infections.
Good friendship, having a good laugh, engaging with somebody is very good for our immune system. Cold showers and cold water swimming, if you can do that. Mediterranean diets or blue zone diets, that's lots of nuts, plants, beans, legumes, fruit, vitamin D. 50% of people in Ireland over the age of 80 are significantly deficient in vitamin D. So take vitamin D supplements, and the dose is 800 to 1,000 international units a day. It works for viral infections, it works for bacteria, and now there's growing circumstantial evidence for vitamin D and COVID.
What makes the TILDA COVID-19 research different from other studies across the world is the second part of the project, which is led by Dr. Nullig Burke.
What we want to understand a little bit more is about how antiviral immunity is activated in older people in Ireland today. Normally when someone's infected with a virus, you activate a very broad, general antiviral immune response. But then over time, your immune system will learn how to educate that response to be specific against a particular virus. And it'll make these things called antibodies. So what we can do is we can measure the levels of antibodies that are specific for SARS-CoV-2 in the TILDA participants and be able to determine who was actually infected with the virus.
The thing about the TILDA study is it's longitudinal, and that is really useful to get a great picture of how people respond to coronavirus. Isn't that right?
Yeah, so what we'll be able to do is, look and see, well, who was antibody positive for the virus in TILDA, and go back and see, well, what are the factors associated with someone having been infected? What are the factors that were associated with someone being infected but being asymptomatic? What were the factors that are associated with someone being infected but developing severe COVID-19? The other really important thing about the longitudinal aspect is that we'll continue to follow these people. If an older person did get COVID-19, what does that mean for their health and well-being in the long term?
We've heard of viruses attacking people and viruses attacking bacteria, but with all this attacking, do viruses ever turn on each other? To answer this, we start with the 1992 discovery, located inside a simple amoeba cell of what at the time was the largest virus known, the mimivirus. Short for mimicking microbe, scientists soon discovered the next size up, the bigger and not to be trifled with mamavirus. This is so big that, just like the Earth, it has its own satellite. And just like the Earth, that satellite virus is called Sputnik. Seriously. Named for the first Soviet satellite sent into space, the much smaller Sputnik satellite virus leaves mamavirus do all the hard work first. That of infecting a host cell, building a viral factory, and setting up a replication assembly line. Sputnik then strikes, hijacking the factory to make more of itself and inhibiting the replication of mamavirus. In fact, so devious is Sputnik that its discovery gave rise to a whole new class of virus, a virophage. A virus that is a parasite on another. In the microscopic world of invasion and infection, satellites and motherships, just like the space race, our understanding of viruses is set to take off. See you next time.
In part one, we looked at the virus that shut down the world in 2020. But despite the negative connotations, not all viruses are bad. Indeed, some are even essential to every single ecosystem on our planet, including our own bodies.
I'm in Cork to meet Olivia McAuliffe, a principal investigator at Teagasc Moorepark.
It's easy to think that all viruses are our enemies, but just in the same way that we have certain good bacteria and bad bacteria, there are also viruses that are actually good for us in that they can promote our health and save us from certain diseases.
What are the viruses that are necessary or good for us?
There's a class of viruses that are called bacteriophages, and bacteriophages are viruses that target bacterial cells only. Phages are everywhere that bacteria are. So that's basically every ecosystem on the planet, in our oceans, in fresh water, in soil, on plant material, and in the human and animal body as well. And they play a very important role there in controlling the microbial populations there. They're completely natural. We coexist with them every day, and they're in fact completely harmless to us.
Bacteriophages are the most abundant and deadly viruses on the planet. First discovered in 1915, they were used to treat infection by disease-causing bacteria. But the arrival of antibiotics in the 30s outpaced phage therapies. It's only recently, with the emergence of antibiotic resistance, that we're seeing a resurgence of interest in phages as therapeutics.
The main difference between antibiotics and phages is that antibiotics tend to have a broad host range in that they kill lots of different types of bacteria, and that can be good if you can't identify the source of the infection, but what it also results in is collateral damage to the good bacteria that are in our body. Phages are very attractive because they're quite specific. So they're specific to the species of bacteria that they infect, and sometimes they only infect a number of strains of that species. So you can use them with precision. They will kill the disease-causing organism while leaving the remaining bacterial population intact. Bacteriophage will dock onto the bacterial cell. They will inject their genome, DNA or RNA, into the cell, and then use the host machinery to produce more viruses. Those viruses then pop the bacterial cell open, killing it, and those viral particles will move on, infecting other cells. And that process replicates over and over again.
Olivia's research focuses on the use of good viruses or phages to control bacteria that cause food poisoning, such as Listeria. Listeria can grow in ready-to-eat foods, such as some dairy products, deli meats and prepared salads, and particularly affects the young, old, immunocompromised and pregnant women. In the EU, the number of Listeria cases is low, but with a mortality rate of 20 to 30%, it presents a high risk to these vulnerable groups.
Listeria, the organism, is very prevalent. It's in the environment. It's everywhere. It's in the soil, it's in water, it's on plant material. It only becomes problematic for us if it enters the food chain and ends up in a contaminated food product. And that generally happens after the product is manufactured and before it's packaged.
PhD student Idelle Stone is working with Olivia to collect and isolate bacteriophages that exist naturally in the environment.
Where do you collect your samples of your bacteriophages?
So wherever the host cell is present really, so different environmental sources such as compost, soil, wastewater, sewage. So I actually took samples from my own back garden, the soil samples, but we were lucky enough to isolate bacteriophage from the grass silage located here in Moorepark. So I isolated 2 bacteriophages and named them after the postcode of Moorepark, so P61 and C996.
So these were phages that hadn't been isolated before?
These were phages that haven't been isolated before, yes.
And you were able to look at them in detail and see how we might use them to tackle something like Listeria?
Yes, so one of my chapters actually focuses on using one of the phages as a biocontrol, so killing the bacteria on different food matrices such as spinach and milk. So that was really interesting and exciting for us to look at.
That is fantastic that out of your PhD you have really concrete findings that are absolutely applicable to everyday lives.
Yes, exactly.
Back in the lab, Adele filters the bacteriophages from the silage and adds Listeria cells before allowing the sample to incubate overnight.
Here you can see it in this image. This is the bacteria growing on an agar plate. And the reason that we don't have a plate here for you to look at is because, again, Listeria is an organism that we would only work with under certain biosafety conditions. And considering that you're pregnant at the moment, we didn't want to expose you to that. So we can see it here on the plate. When we add the phage sample in there, the phage will kill the bacterial cells, and wherever the phage have killed the bacterial cells, you will see a clearing on the plate. So that's what we call a plaque, and in that plaque are the live viruses. So we can then go to the plaque and purify viruses from there.
So when you say purify them, you want to just take the phage out and really look at how exactly it works?
Yes, part of the work that we're doing and that Adele is doing is we're looking for a broad host range phage. So that's a bacteriophage that's going to kill lots of different strains of Listeria monocytogenes. Because if you've got Listeria monocytogenes in your food product, you're not going to know which strain of Listeria monocytogenes it is. So you want a broad host range phage that's going to kill that Listeria no matter what the strain is. Our approach is to use these phages in the food production facilities. So on the surfaces, the stainless steel surfaces, on the floors, in the drains, on the walls. In most modern production facilities, there's very rigorous cleaning regimes anyway, and they would be designed to remove Listeria. So what we're doing is trying to give them another level of protection that's Listeria specific and could be used in places where Listeria is particularly problematic.
So with these phages that you would be almost putting onto a food, does that have a connotation for the public to, be very scared of because you're putting a virus onto something to try and get rid of another bacteria?
That's certainly true, but with any approach to controlling disease-causing bacteria, there has to be a level of education and awareness of the public. So just like there has been a public acceptance of good bacteria versus bad bacteria, I think we can educate the public to show them that there are also good viruses versus bad viruses.
So where a virus was the villain of 2020, in the future they might actually be our heroes.
That's right, and we're looking to harness their killing potential to tackle harmful bacteria.
That's our 10 things to know about viruses, and it wraps another fascinating series of science research. Trust in science has never been so important, so it's great to look ahead knowing that through Ireland's research community, we're in good hands.